So my presentation is on IPS, that is induced pluripotent stem cells, and how to reproduce without gametes. So to begin, what is a pluripotent stem cell? Well, pluripotent stem cells are cells that we can take from embryos. We normally use to take them from embryos, and we generally use them to generate almost any tissue. Wait, is my presentation loading? Yes, it is. Okay. So where do we take them? Those pluripotent stem cells. We take them from embryos. So we need to have a sperm cell and an egg cell. We have to do the fertilization. It has to go to zygote, second cell stage, fourth cell stage, morula, blastocyst, and then the actual stem cells come from the inner cell mass, which we call embryoblast. And those embryoblasts, they can become any cell in the body, any human cell, they can differentiate into. But there is an issue using those stem cells, embryonic stem cells. The first issue is it ethical to use embryos as a resource? Because when we take those stem cells from embryos, we use those embryos and they cannot be used to generate a new life. We can use them only once. We can take as many cells from the inner cell mass for the embryoblast as we like, but the embryo will not will not be able to form a new human being after this. Also, there is a problem that it takes a long time to establish a cell line. It goes from 6 months to an entire year just to establish a single cell line. And that technology also has an issue because it kind of facilitates human cloning and designer babies, which is a whole other ethical issue. It's really complex. But so what's the solution? Oh, actually, there is a Japanese named Shinya Yamanaka. I hope I said this right. He won a Nobel Prize for this discovery: the induction of pluripotent stem cells from mouse embryonic and adult fibroblast cultures by defined factors. So, what does all of this mean? What he did is he took fibroblast cells. Fibroblast cells are like skin cells. You can take cells from skin, and they did that with mice, obviously. And you can add some factors, some transcriptor factors, and they can reprogram those cells for de-differentiate them. That is, turn them back into the state of a stem cell. So they can reprogram those cells, and then use those cells that used to be skin cells to make any cell in the human body, because they have become stem cells. And how do we make use of this technology? Well, the theory is if we can take fibroblasts, skin cells, and then use those factors to reprogram a cell, we can de-differentiate them, do a culture on a specific cell, and then use those different cells from a patient to do drug screening, disease modeling, use cell-based therapy, and it won't have any rejection because they have the same DNA of the person. So there is no incompatibility. And putting it a bit from my side, which I work with, I study human-assisted reproduction. What could this mean for reproduction? In reproduction, we have people that cannot produce gametes. They can produce sperm cells or egg cells by a huge number of factors. But this could mean that we can take some skin cells use those skin cells, reprogram them, and make new gametes. So we can, for example, a male patient that can produce sperm cells. We take some skin cell, reprogram those cells, and then we can have sperm cells. It doesn't produce, but we just made them in lab. That is really an amazing thing to do. And then we can take those sperm cells and use it in the in vitro fertilization, which is a technique for making embryos in laboratory. And then we can transfer them to a female patient, or for the couple. Anyways, for the patients who are seeking treatments in human assisted reproduction. Here there is another example. We have a male donor, a male IVF donor. IVF is in vitro fertilization. We take those cells and make it gametes. But there is a few problems with this. So one of the things that we can also it's theorized that we can also do is we can take cells, we can reprogram them using IPS technology. We could make artificial gametes production of different cells. We could make an egg cell from a male. So this is quite a bit of a problem, because then we could do a self-fertilization. And that leads me to the issues. There are some risks in using IPS cells. One of them is the safety, because there is a risk of them turning cancerous, because it's quite hard to control how much they divide, how much they spread. So this is a risk of turning into tumors. And also for the reproduction, like this, would we be cloning a person if we take a sperm cell and an egg cell made from cells of the same person? Wouldn't that be cloning? And so we would be creating clones. So that would be a whole other ethical issue. But there are some benefits. What are the benefits of this technique? So in human reproduction, there is a problem with the aging of women, because when women are 35 years plus, the egg cells are not the same quality as they used to be. The quality drops like tremendously. But if we used the IPS cells, it would probably be expensive, yes. But if we could use the IPS technology to make those new egg cells, they would be new. They wouldn't be of a 35-year-old woman, because they would be differentiated from a stem cell. And the quality would be preserved as if it was a new cell. Also, there would be no need for hormone therapy, because when a female goes to the reproduction clinic, there is a need for them to take some hormones for them to produce egg cells in mass. So, and those hormones, they can be quite intrusive. They can influence a bit on life. But if we use this technique and we use, for example, the skin cells, it wouldn't be any need for this. There wouldn't be any need for the hormone therapy. And also, there would be no need to freeze the cells. To freeze gametes, we could just take them on the go. You go to the clinic, we take a few skin cells, we send it to a lab that does IPS, or we could do IPS on the clinic. and then we would do the fertilization process in vitro with almost new cells, practically new cells. So it would be really interesting. So yeah, there are a lot of ethical issues with IPS still, but I think that with further discussions in the scientific community, we could break this barrier and make it more noticeable and more worldwide knowledgeable that this is a possibility. We can actually take a cell from anywhere of your body and make it another cell that you if you need any other cell, we can make that cell. That's what I'm trying to say. Also, I did forget to mention, but this discovery, when Yamanaka won the Nobel Prize, he won it in 2012, and the research that he did was all the way back in 2006. So we are almost 20 years on this discovery, and I noticed that not a lot of people do know about this. So that's the main reason why I wanted to present it as well. And yeah, that's pretty much it. It was a little bit quicker than I expected, but yeah. Thank you very much.
Okay. So, now that the presentation is done, we are once again open for any questions on the subject.
Yeah, so someone else had a question in one of the chats on CRISPR.
CRISPR. Yes, CRISPR is a gene editing technique. It is quite interesting as well. Are they asking what specific about CRISPR? I
just have to say, like, I kind of rolled my eyes a little IRL because when you're actually into in the field of biology and genetics, CRISPR is like so yesterday's news at this point.
Oh.
It's not as good as we were hoping it would be.
Yeah, it's not as
and
good.
there are better techniques now.
I
Yes.
think the question was just if we were going to discuss CRISPR at all. But yeah,
Oh,
CRISPR
no.
Was like from a while ago, right?
Yes,
So
CRISPR.
what is the help enlighten us, really? What is the cutting edge on science now?
There's like a bunch of different techniques.
There are a lot of techniques. Oh, CRISPR is still good. It's still good.
It's not abandoned. It's still good. You can't use it for everything because it has drawbacks that we've discovered. There are a bunch of other techniques we also use alongside CRISPR.
Indeed.
Do you have any examples?
Not off the top of my head. I don't do direct genetic engineering stuff.
Yeah, I don't work a lot with CRISPR as well. But
that's not
CRISPR. When was CRISPR discovered? It was quite a while ago.
CRISPR like 10 years ago?
Yeah, I think something like that.
Yeah, I remember.
I know it was like a while ago, and then like it isn't until like recently that people really were trying to do a lot of stuff with it, like in the past couple of years.
I do see a question in a chat from BitShepherd asking, the sort of engineered gametes that you were discussing, are those being done in animal models yet?
Yes, they have been done in mice, and they have actually that thing when we can take a cell from example a male and make both a sperm cell and an egg cell. They have done it with a mice, and they have fertilized. They have united those gametes, made an embryo, and that embryo was yes, that embryo has hatched and it become a new mice, and it was a live mice, a healthy mice.
Oh wow. I know, like.
Oh yes, one of the new big ones is prime editing, which is a fancy suite of RNA and protein, especially designed RNA and protein complexes that let you replace individual base pairs in situ. That's the big new thing.
I haven't heard of it yet. I might do a research after.
There is
That's like 2021, 2022 that started like becoming a thing, and it's a real big thing now.
yeah. Someone in my question has a more kind of different kind of question.
Yes, I will be
Asking
talking about
who-
epigenetics.
Oh no. Above that, do we know whatever happened to the Chinese newborns who had work done on them by a rogue doctor?
No, we don't.
Yeah, I don't know what happened
No,
to
we
the
don't,
twins.
and it's questionable whether he was telling the truth when he said he did it.
Well, wasn't he like banned or something of the
Yeah.
science
He
field?
was stripped
Largely
with like
banned.
he was kicked out of all the professional organizations he was a part of, stripped of all of his licenses to practice and
Yes.
stuff.
Yeah.
Yeah.
That's
We
crazy.
don't know if it's questionable whether he was even telling the truth about having done so.
Well, I don't believe it. It would be impossible. It would be possible to do it. What he said he did, but
He said he did it, but even if he did, like, how would you test that? It wouldn't be
yeah,
like,
yeah.
it wouldn't be ethical to test that.
Well, it wouldn't be ethical to do it in the first place, but okay.
Yeah, it wouldn't be
Yeah.
ethical to do it, and it wouldn't be ethical to test it. It's like test if it actually worked. It's such a huge problem.
So either he did it and should have lost all his credibility, or he lied and should lose all of his credibility. So
Yes.
Yeah.
whether or not he lied or not, we did the outcome kind of is the same.
It's the same.
Yeah.
It's true.
Yeah, we have learned a lesson. Please don't do gene
Experiment on
editing.
humans.
Don't do experiment in humans without their consent. Please.
Right,
Yep.
exactly.
Yeah.
Informed consent
And
is an
without
important
like
thing.
other testing on like, animals, and even before animals, like actual testing beforehand, like anything besides just starting with humans. That's crazy.
And that's why we have councils for that.
Mhm. Turns out going absolutely rogue will get you to lose your job. But anyway,
Especially in these fields.
Mhm.
okay.
I got a question.
Sure.
he-
Can this fix my receding hairline, and if so, how soon? That is a legitimate question, though, but I did want that as a
Well,
joke.
there is a few research and application of it. There aren't many applications yet, but the thing is, we need to take care when we use it to do it in humans, because there is like there is a I don't know if it is how small or how big it is, but there is a chance of developing into cancer. So yeah, probably not
Strictly
not the best of
speaking,
ideas.
This would be an application of the bioprinting of skin and epidermal tissue, like in tissue engineering, which is a bit above the scope of specifically talking about stem cells.
Yes.
But it's definitely an application. If we get to the point where we can just like make skin from scratch, because we could also make scalp skin with the hair cells in it and surgically implant that.
Sounds exciting.
Yeah.
I do love the research on stem cells and tissue generating. Really dope.
Oh yeah,
Okay.
that could revolutionize.
There will probably be
whole
a
bunch of
pill you
medicine.
can take to stop your hair loss before that is a thing, though.
Yeah, probably.